Good morning, everyone. I'm delighted to welcome you all today. Today, we will be exploring the topic of prospective validation in semi-solid and liquid formulations. My name is Dr. S.K. Singh, and I hold a PhD in this field. This presentation will cover key aspects of validation processes, best practices, and recent developments. I look forward to sharing insights and engaging with you throughout this session. Let's get started. Prospective validation is a crucial step that we perform before we move into commercial manufacturing. It helps us demonstrate that the process we use for semi-solid formulations, like creams, gels, and ointments, as well as liquid formulations such as syrups, suspensions, and solutions, is truly capable of producing consistent results. This means verifying that the process is reproducible, so it can be reliably repeated and controlled, meaning we can manage and maintain the quality throughout production. The key goal here is to ensure that the final product consistently meets the defined critical quality attributes, or CQAs, which are the specific properties that determine the quality and effectiveness of our product. By doing this validation upfront, we can confidently say that our manufacturing process is robust and reliable, which is essential for maintaining product quality and ensuring patient safety. This step aligns with our overall theme of quality by design and helps us build a solid foundation for successful commercial manufacturing. Today, I want to talk about the main objectives of prospective validation. First, it's about establishing documented evidence that our manufacturing processes consistently produce high-quality products. This means we need solid proof that what we make meets our quality standards every time. Next, it's crucial to verify that our critical process parameters, or CPPs, stay within their defined limits to ensure our critical quality attributes, or CQAs, are consistently achieved. In other words, we want to be confident that the process controls are effective and stable. Another key point is eliminating process variability and confirming reproducibility. We do this through 3 successful validation batches, which demonstrate that our process is reliable and produces the same quality product each time. Finally, we must ensure our validation efforts meet regulatory expectations. This means complying with the requirements set by agencies like the FDA, EMA, WHO, and PICS overall. These objectives help us maintain quality, consistency, and compliance throughout our manufacturing process. Now, let's take a closer look at the various product types we encounter in our field, including lotions, creams, gels, syrups, and suspensions. Each of these has unique characteristics that influence how we develop and manufacture them. When it comes to API characteristics, key factors such as solubility, stability, and viscosity impact not only the formulation process, but also the final product performance. For example, solubility determines how well the active ingredient dissolves, while stability ensures the API remains effective over time. Viscosity influences the product's spreadability and user experience. Excipients play a crucial role in formulation as well. Preservatives are essential for microbial stability. Emulsifiers help blend ingredients that normally don't mix well, and stabilizers maintain the integrity of the product over its shelf life. Moving on, the Quality Target Product Profile, or QTPP, serves as our blueprint for quality. It defines what we aim to achieve in terms of safety, efficacy, and quality attributes. From there, we identify critical quality attributes, or CQAs, which are the essential properties that must be controlled. These include pH, which affects stability and skin compatibility, viscosity and rheology, which influence application and feel, globule size or particle size, especially in suspensions, impacting stability and bioavailability, assay and uniformity to ensure consistent dosing, microbial quality for safety, and appearance and clarity, which affect consumer acceptance. Understanding both the product and the process is fundamental to successful formulation and manufacturing. It helps us predict how changes might affect quality and performance. Finally, key components of prospective validation include establishing robust processes that consistently produce a product meeting all quality criteria. This proactive approach ensures that from development through production, we maintain high standards and deliver reliable, safe products to patients. Now, let's take a look at some of the typical risks we encounter in this process. These include variations in mixing uniformity, which can affect the consistency of the final product. Emulsion stability failure is another concern, as it can lead to separation or degradation over time. Inadequate homogenization might result in uneven distribution of ingredients, impacting efficacy and quality. Preservative ineffectiveness can pose safety issues, making it crucial to monitor and validate their performance. pH drift and temperature-sensitive degradation are also risks, since fluctuations can compromise stability and potency. Additionally, microbial contamination in aqueous systems remains a persistent challenge, especially for products with high water content. All of these risks highlight the importance of identifying the critical material attributes, or CMAs, and critical process parameters, or CPPs. By doing so, we can better control and optimize the manufacturing process. To systematically evaluate these risks, we often use tools like failure mode and effects analysis, or FMEA, Ishikawa diagrams, and HACCP principles. These approaches help us prioritize risks and develop robust mitigation strategies. In the context of our overall validation efforts, understanding these key risks and their management forms the foundation of prospective validation. This process ensures that our manufacturing process is reliable, consistent, and compliant with quality standards, ultimately safeguarding product quality and patient safety. Let's take a closer look at what makes a well-defined protocol. First, it clearly states the scope and purpose, setting the boundaries and goals for the validation process. Next, it includes a process flow diagram, which visually maps out each step involved. Detailing the equipment and facilities used is also crucial, ensuring everything is suitable and properly qualified. When it comes to batch size and the number of batches, typically 3 full-scale batches are used to demonstrate consistency. The sampling plan and specific locations for sampling are outlined to ensure representative data collection. In-process parameters and their acceptance criteria are defined to monitor quality throughout the process. Analytical tests are specified to verify the product's quality at different stages. Handling deviations and obtaining approval for any unexpected issues is another key component. It keeps the process controlled. Statistical evaluation methods are employed to analyze the data objectively, confirming the process's robustness. The validation protocol itself, often called the VP, ties all these elements together into a comprehensive plan. Understanding these key components is essential because they form the foundation of prospective validation. They help us ensure the process is reliable, consistent, and compliant with regulatory standards. This structured approach ultimately supports the overall quality and safety of the product we're bringing to market. Now, let's dive into some of the key steps involved in developing and validating our semi-solid formulations. First, when adding heat-sensitive ingredients, we need to be very careful to preserve their stability and activity. Deaeration is also critical here. Removing air helps prevent oxidation and ensures product quality. Next, during filling and packaging, controlling pump speed is essential to maintain accuracy and consistency, while ensuring tube integrity or jar filling consistency helps us avoid leaks or uneven filling. Moving on, melting and phase preparation involve carefully heating the oil and aqueous phases. Temperature control and hold time are vital to achieving the right phase behavior. During emulsification, we need to pay attention to the phase addition rate, as it influences the stability of our emulsion. Agitation speed and duration are also key. Too fast or too slow can impact quality. Homogenization, pressure, and cycles further refine the emulsion, making it uniform and stable. Once the product is formed, cooling and viscosity development are next. A controlled cooling rate helps us prevent defects like separation or crystallization. Shear impact during cooling can influence viscosity, so managing shear is important for the desired texture. Finally, all these unit operations are interconnected, and their proper validation is crucial. Validating each step ensures we consistently produce a high-quality, stable semi-solid formulation, aligning with our overall goal of reliable product development. Let's delve into some critical process parameters that influence our formulation quality. First, agitation speed is vital because it affects mixing efficiency and uniformity. Adjusting the temperature set points ensures the right environment for reactions or phase changes, impacting stability and consistency. Homogenization pressure and time are also key. They determine how well the components are blended, especially in semi-solid formulations. The phase inversion point is another crucial concept. It marks the transition where the emulsion changes from oil in water to water in oil, which can significantly impact product performance. The mixing order and addition sequence are often underestimated, but are essential for controlling the formation and stability of our formulations. Proper sequencing can prevent issues like phase separation or incomplete mixing. All these factors tie into what we call critical process parameters, or CPPs, those variables we must control tightly to ensure quality. Finally, when we talk about prospective validation for semi-solid formulations, we're essentially planning ahead to verify that our process consistently produces a product meeting predefined quality attributes. This proactive approach helps us ensure robustness and reliability in manufacturing. All these points are interconnected and form the backbone of a well-controlled, high-quality formulation process. Let's delve into some critical quality attributes for semi-solid formulations. First, viscosity plays a key role in determining the application and stability of the product. It's essential to control it to ensure proper spreadability and user experience. The rheology profile helps us understand how the material flows and deforms under stress, which impacts both manufacturing and application. Next, globule and particle size are crucial because they influence the texture, stability, and drug release profile of the formulation. pH is another vital parameter. Maintaining the correct pH ensures compatibility with skin or mucosal tissues and stability of the active ingredients. Spreadability is about how easily the product can be applied uniformly, impacting patient compliance. Assay and content uniformity are fundamental to guarantee that each dose contains the correct amount of active ingredient, ensuring efficacy and safety. Microbial limits are critical too, especially for semi-solid products, since they are often applied to sensitive areas, and we must prevent contamination. Stability performance assesses how the product maintains its quality over time under various conditions, which is essential for shelf life determination. These are all considered critical quality attributes, or CQAs, for semi-solid forms. Finally, when we think about prospective validation, it's about establishing robust processes that consistently produce products meeting these CQAs. Validation ensures that throughout manufacturing, from raw materials to finished product, we maintain quality standards. This comprehensive approach ultimately supports product efficacy, safety, and regulatory compliance. Let's take a closer look at some key unit operations involved in liquid formulation development. First, for emulsions like lotions, the process begins with oil-water phase preparation. This step is crucial because it sets the foundation for a stable emulsion. Next is emulsification and homogenization, where we ensure the oil and water phases are thoroughly combined to create a uniform, stable product. The effectiveness of stabilizers also plays a vital role here, helping to prevent separation over time. Moving on to suspensions, the focus is on wetting of the active pharmaceutical ingredient, or API. Proper wetting ensures the API disperses well within the liquid medium. Particle size control is another critical factor. Smaller, uniform particles tend to stay suspended longer and provide better consistency. Uniform mixing and homogenization are necessary to achieve a homogeneous suspension, avoiding settling or segregation. And speaking of settling, understanding sedimentation volume helps us predict how long a suspension remains stable under storage conditions. Finally, solutions like syrups and oral liquids involve solubility optimization, which is all about ensuring the drug dissolves properly. Heating and mixing are often used to facilitate this process, making sure everything dissolves evenly. Filtration and deaeration are also important steps. They remove impurities and entrapped air, which can affect the product's stability and appearance. All of these steps are interconnected and vital for the prospective validation of liquid formulations, ensuring our products are safe, effective, and consistent for the end user. Now, let's take a closer look at some of the critical process parameters that are essential for ensuring the quality and consistency of liquid formulations. First up is mixing speed and time. These factors are crucial because they directly influence the uniformity of the mixture, ensuring that ingredients are evenly distributed. Next, temperature control plays a vital role. Maintaining the right temperature helps preserve the stability of sensitive components and supports proper chemical reactions during processing. Homogenization shear rate is another important parameter. It affects the particle size and dispersion, which in turn impact the product's stability and performance. Moving on to filtration specifications, these are key to removing any unwanted impurities and ensuring clarity and purity of the final product. Transfer process controls are also critical. They help prevent contamination and maintain process integrity as the product moves between different stages. Lastly, monitoring pH and preservative effectiveness is essential for ensuring microbial stability and extending shelf life. All of these parameters are interconnected and must be carefully controlled to validate the consistency and safety of our liquid formulations. This is where prospective validation comes into play, allowing us to predict and verify that our process will reliably produce high-quality products every time. Let's take a closer look at the critical quality attributes, or CQAs, for liquid forms. First, pH is crucial because it can affect the stability and solubility of the formulation, as well as its compatibility with the body. Next, density or specific gravity helps us understand the formulation's flow and mixing characteristics, which are essential for consistency. Viscosity is another key parameter. It influences how easily the liquid can be administered and how it behaves during storage. When dealing with suspensions, particle size becomes important because it impacts stability and uniformity. Smaller, well-dispersed particles help prevent sedimentation. Assay and uniformity are fundamental to ensure each dose contains the correct amount of active ingredient. Clarity, especially in solutions, indicates the absence of particulates and impurities, which is vital for patient safety and product quality. Sedimentation characteristics are also significant in suspensions, as they affect how quickly particles settle and how easily the product can be redispersed. Microbial limits and preservative efficacy, often assessed through preservative efficacy testing or PET, are critical to prevent microbial contamination and ensure the product remains safe during its shelf life. Now, connecting this to the overall theme, understanding these CQAs helps us ensure that our liquid formulations are not only effective, but also safe, stable, and of high quality through proper validation processes. Prospective validation, in particular, allows us to predict and control these attributes before production, reducing risks and ensuring consistency downstream. Let's talk about phase temperature and mixing uniformity first. Ensuring the right temperature during each phase is crucial for product stability, and uniform mixing helps achieve consistency throughout the batch. Homogenization efficiency is next. This process is vital for breaking down particles to create a smooth, stable product, which directly impacts quality. Weight and fill volume control are also key. Precise control here ensures each package contains the correct amount, maintaining compliance and customer trust. Monitoring pH and viscosity is equally important, as these parameters influence product performance and stability, so continuous tracking helps us catch any deviations early. Emulsion and suspension uniformity are critical, especially for complex formulations. We need to make sure these are consistent to avoid separation or instability later on. Microbial control is another fundamental aspect. Preventing contamination ensures safety and extends shelf life. Line clearance and equipment cleanliness are often overlooked, but are essential for preventing cross-contamination between batches. This is where in-process controls, or IPC, come into play. They allow us to verify that each step is performed correctly, ensuring the integrity of the final product. All these points are interconnected. They form a comprehensive approach to maintaining high quality, safety, and efficiency throughout production. By focusing on these aspects, we can confidently deliver products that meet both regulatory standards and consumer expectations. All right, now that we've established the initial process setup, the next step is to conduct 3 consecutive commercial-scale batches. This is a crucial phase because it demonstrates the reproducibility and reliability of the process at a larger scale. We want to ensure that everything runs smoothly across multiple runs, not just a one-time event. It's important that these batches are handled by trained operators using qualified equipment, which means all the IQ, OQ, and PQ protocols have been completed beforehand. This ensures that the environment and equipment are properly validated and ready for commercial production. During these batches, we meticulously record all process parameters, deviations, and adjustments. This detailed documentation is key because it allows us to track exactly what happened during each run, identify any issues, and make data-driven decisions for continuous improvement. We also perform complete sampling according to our established protocol. This means taking representative samples at specified points to analyze the quality and consistency of the product throughout the process. Finally, we evaluate the batch consistency through statistical analysis. This step helps us confirm that the process is stable and capable of producing product within the desired specifications consistently. All of these steps together form the core of our validation execution, ensuring that our manufacturing process is robust, reliable, and ready for full-scale production. Let's talk about compliance with all critical quality attributes, or CQAs. It's essential that we meet these to ensure product quality and safety. I'm pleased to report that there have been no critical or major deviations, which indicates our processes are well controlled. Our critical process parameters, or CPPs, remain within validated ranges, confirming that our manufacturing process is consistent and predictable. When we look at batch-to-batch consistency, we see stable key parameters such as viscosity, which affects how the product flows; phase stability, ensuring the product remains uniform over time; particle and globule size, which impacts safety and efficacy; pH, critical for stability and activity; assay results, confirming the correct dosage; microbial results, ensuring the product is free from contamination; and packaging integrity, which must meet our specifications to protect the product until use. All these factors are evaluated against strict acceptance criteria to maintain quality standards. Overall, maintaining this level of consistency and compliance is fundamental to our commitment to delivering safe, effective, and high-quality products. Now, let's look at the key elements involved in the final stages of our validation process. First, we have protocol approval, which confirms that all procedures and plans meet the necessary standards before moving forward. Next, we review batch records and IPC data to ensure that each production batch aligns with the approved protocols and quality requirements. Lab test results are then evaluated to verify that product specifications are consistently met. Equipment qualification summary is also crucial, as it confirms that all equipment used in manufacturing and testing is properly qualified and functioning correctly. We then examine deviation reports and CAPA, or corrective and preventive actions, to address any issues that arose during the process and ensure they are properly resolved. A statistical evaluation of data trends helps us analyze the data comprehensively, identifying any patterns or anomalies that could impact quality. Once all these aspects are thoroughly reviewed, we compile the final validation report, summarizing our findings and confirming that the process is validated. Finally, we organize all validation documentation, ensuring that every piece of information is properly documented and accessible for future audits or reviews. These steps collectively ensure that our validation process is complete, robust, and compliant with regulatory standards, reinforcing our commitment to quality and consistency throughout the product lifecycle. Now, let's talk about prospective validation and why it's so important. It ensures that our semi-solid and liquid formulation processes are scientifically robust, meaning they're based on solid scientific principles. It also makes sure these processes are reproducible and controlled, so we can consistently produce the same high-quality product. Importantly, it aligns with regulatory requirements, helping us stay compliant and avoid issues down the line. Ultimately, prospective validation confirms that we can reliably deliver products that are safe, stable, and of the highest quality. So, in conclusion, implementing prospective validation is key to maintaining process integrity and product excellence throughout our development and manufacturing stages. For further support, please don't hesitate to reach out. We're here to help you with any questions or assistance you might need moving forward. Thank you so much for your time and attention today. Your engagement is truly appreciated, and I look forward to connecting with you soon.